welcome Dr. Clive Bramham. He's a professor and head at neuroscience at the Department of Biomedicine at the University of Bergen, Norway. His lab elucidates the cellular and molecular mechanisms that control synaptic plasticity and the dysregulation of these mechanisms in cognitive disorders. He's made seminal contributions to the discovery of BDNF as a trigger for transcription and translation-dependent synaptic activity in the adult brain. And currently he is the president of the Norwegian Neuroscience Society and a board member of the Norwegian Research School in Neuroscience. Thank you very much for being here.
Thank you very much. First, I'd like to thank Mike and Tu and Hal for putting together this fantastic meeting. This talk will be about activity-dependent synaptic plasticity and its molecular control. And specifically, I'm focusing on long-term forms that depend on gene expression and regulated protein synthesis. And more recently, we've become intrigued by the prospect of being able to image specific forms of plasticity as they take place at synapses and potentially to interrupt specific forms of plasticity at the level of expression. And I'd just like to trace the path that's taken us there so far. I think that this talk relates quite closely to what Errol was saying today, Michael and Karel, as well as Anders yesterday. So we've heard quite a bit about the basics of synaptic plasticity already, and the idea being that behind long-term behavioral change, activity-driven changes of one form or another of synaptic plasticity, that they play a role, they play an important role. It's studied throughout the brain and spinal cord. It's a widespread, ubiquitous phenomenon at glutamatergic synapses, although it's highly diverse. Normally, you think of synaptic plasticity in terms of long-term memory formation, and there's been more and more interest recently in how perturbations in synaptic plasticity and perturbed homeostatic regulation may contribute to a span of neuropsychiatric disorders, cognitive dysfunction. But really, not much is known about the specifics of that sort of dysfunction. And so if you look at being activity-dependent forms of plasticity, you have LTP and LTD are frequently referred to. These are very generic terms. You really can't say that because there's so many different flavors of LTP and LTD. Depends on how it's induced and what brain region you're talking about, and as we heard earlier, also the specific synapse and its prior experience. And there's also a homeostatic regulation that takes place. So in each of these cell types within the hippocampus, for instance, their morphology is obviously very different. But it's also true at the level of the synapse, at how the postsynaptic density is constructed, and how the synaptic plasticity plays out within this circuitry actually differs. And I'll show you some evidence of that. So there's a lot of diversity, and we know that in phenomena such as LTP, which was discovered by Terje Lermo in Oslo, and then really developed by Tim Bliss and Terje Lermo at the National Institute for Medical Research in London, for which Tim won the Brain Prize. So LTP, it does involve usually stable structural changes, regulation of the actin cytoskeleton. But this, you're getting a bigger synapse, you have to have more of the constituents of the synapse. It's not really known exactly how that's being organized. But key to this, a key aspect of this is the local translation of messenger RNA in dendrites. So some 10% of the transcriptome is actually present in the dendrite to begin with, starting out. As we'll hear about later, it's also a really interesting messenger RNA that is produced on cue, on demand, and rapidly delivered out into dendrites for local translation, and that is ARC, immediate early gene product. So looking at these dendritic protein synthesis, we were interested in really the biochemistry of how this was taking place in the neuron and in dendrites. And so looking at sort of textbook view of the dogma of molecular biology, you get to this point here, and so the main determinant of protein levels is really at the level of translational control and protein degradation as well. And so we wanted to know more about this process. So then you get then into a number of complicated pathways, effector systems for regulating protein synthesis. A canonical pathway is regulated by the mammalian target of rapamycin. It regulates pretty much general levels of protein synthesis. It's strongly implicated in that. And so what we found when we looked at long-term potentiation, the dentate gyrus of the adult rat, is that actually this mTOR pathway does not figure into the mechanism. And what we found is the makings of a new mechanism that's mediated by ERK signaling to MAP kinase interacting kinase, MNK. And so I'll just, it's not going to be heavy on data. I'll just try to point to some of the highlights here. And so what Deborah Panya did, a postdoc in the lab, he looked at the role of BDNF in this LTP in the dentate gyrus. It's in a live animal, an anesthetized animal. And when he applies the BDNF scavenger TRAC-BFC, and you can see that when applied at different times, it will completely and permanently revert the LTP in a time-dependent way. So when you get out to 8-10 hours, the effect of the TRAC-BFC disappears. So there's a window of BDNF TRAC-B signaling that's driving this process of stabilizing LTP, consolidating LTP in the dentate gyrus. And he showed that if you block the mTOR pathway, it doesn't do anything to this LTP, no matter when it's applied, high concentrations and so forth. And we were able to show that the drug is working, it's blocking mTOR and downstream signaling, no effect on the LTP. But what he sees, though, what he finds is a role for this MAP kinase interacting kinase, and it is regulating a more specific form of translation that's under the control of Fragile X mental retardation protein. The loss of this protein is a common cause of intellectual disability. And so how this works is for translation to get started, which is the most highly regulated step in protein synthesis, the messenger RNA has to engage the ribosome. And that's controlled at the level of this initiation 4E binding protein. Anyway, FMRP and this protein CYFIP, they block that interaction. And he showed that during LTP, CYFIP and FMRP are released, and then the targets, the messenger RNA targets of FMRP undergo enhanced translation. And so just to simply put, it's like this, and it's all dependent on TRAC-B signaling and to the MNK pathway. And it divides into two stages, and so, such that it looks like there's a translational program that's playing out in the consolidation of the LTP. So it starts with the early translation of the FMRP targets, and then it switches to a mechanism that we can only detect biochemically within the fractionated synaptoneurosomes. We've done this in various ways, in vivo and in vitro stimulation, also using MNK1 knockout mice, pharmacological inhibitors, and so forth. So it has the makings of a translational program, a switch from specific translation to more general dendritic protein synthesis. Okay, and for the work of Chris Proud, he has also obtained evidence in a number of other labs that this MNK pathway, which phosphorylates initiation factor 4E, is also involved in the regulation of the epithelial mesenchymal transition in cancer metastases. So there's really interesting parallels, although the FMRP targets are different in the cancer situation, if you will, and synaptic memory, synaptic plasticity. Okay, and I'd like to turn now to ARC. Which is, to us, a highly intriguing specialized system for information encoding in the vertebrate brain. It's hair-triggered. The ARC is expressed in projection neurons in vertebrates. The POL2 is stalled on the start site, so very rapid transcription. The messenger RNA, a good portion of it, is super rapidly trafficked out into dendritic processes. As the ribosome moves along and you start making the protein, the messenger RNA undergoes a very rapid decay. It's because there are these two introns in the 3' UTR I won't go into the detail. The messenger RNA is rapidly turned over, and the protein can also be very rapidly degraded by the UPS system, by ubiquitination and proteasomal degradation. And then when you have the protein, what's really interesting is that the protein is an indispensable component for LTP, LTD, as well as homeostatic scaling, as well as in long-term memory, and in the critical period in the visual cortex. So this is then an ARC-centric view of things, where ARC plays a critical role in regulating these forms of long-term plasticity, long-term synaptic plasticity, and we know that it's involved in memory and developmental aspects, plasticity as well. Okay. So just to show a bit of data, so here you see the LTP that's induced by brief bursts of high-frequency stimulation to the perforant pathway. Here we've induced it by local infusion of BDNF into the dentate gyrus, and we've applied an ARC antisense, which then targets and inhibits specifically the translation just of ARC now, and you'll see that ARC really is important for this consolidation process. And this is, again, very time sensitive. So if you apply the ARC antisense now later on, it does nothing to the maintenance of this long-term potentiation. Okay, so we wanted to then know how ARC is involved in these different types of plasticity. I mean, there are many possibilities, but we do think that it has to do with regulation of the ARC protein itself. Okay, a post-translational modification, a switch, a property of the ARC protein that underlies this. And so our thinking is something like this. You have these different types of plasticity. ARC is a form of master regulator, and it's evolved then in this, obviously functionally important as well as in diseases. And I think that this notion was really underscored by a series of papers, and particularly these last 3 papers related to schizophrenia and autism spectrum disorder, where they showed that that proteins that are connected to ARC in the ARC interaction network, they are highly enriched for rare mutations and copy number variations in schizophrenia and in autism spectrum disorder. ARC, no. We've looked at ARC ourselves in collaborations with geneticists and have not seen so far really anything significant in the ARC gene itself. So there's a network around ARC, and this then is the ARC protein interaction network in the human postsynaptic density. Just to show the hubs and to highlight the massive interactivity of ARC with other functions of the PSD and the neuron. This was made by Ian Simpson at the University of Edinburgh. And so we've done biochemistry over the last few years. And you see this is ARC then in the center, and these are ARC interacting proteins. Several of these proteins have been quite well shown to directly bind to ARC. These are from the literature, and these are proteins that we've found by either using GST pull-down or by co-immunoprecipitation, and these are the members of the lab that have been doing this. And so we're following up then a number of these novel proteins in terms of their function. And interestingly, one of them is actually ERK, and we see that ERK does phosphorylate ARC. It's a paper that we're submitting pretty soon. So if you look then at the ARC interaction network, some of it nicely scales down into these pathways. So ARC is involved in endocytic trafficking. In LTD, ARC is thought to act as adapter protein for endocytosing glutamate AMPA receptors and weakening the synapse in that way. It's also involved in the processing of APP to beta amyloid, interestingly enough. It regulates the cytoskeleton, but we don't know exactly how. And it's really important to note that ARC also enters the nucleus, and several different roles of ARC in the nucleus have now been described. So, so we really wanted to go further because really nothing is known about the protein itself. What does it look like? What sort of shape does it have? And we needed to know this. And so we started a collaboration with Aurora Martinez and several other people, put a team together and to look at the properties of the ARC protein. And it's 20 years since ARC was first found. And this is the first work looking into the basic biophysical and structural properties of the bacterially expressed human recombinant ARC protein. So Craig Myram, a PhD student, he managed to purify it, and then a number of different analyses were performed. And basically what came out of it is that we were able to provide evidence that it has two major domains and an unstructured linker region. And throughout the domains, it is a flexible, floppy protein in terms of its properties. We also show that the recombinant protein exists as a monomer and undergoes reversible oligomerization. So with high temperature, you can get it to oligomerize and bring it back down. This shows some results from the dynamic light scattering. And here you can see the monomer, and this may be either an elongated monomer or a dimer. We're not exactly sure of the oligomeric state of ARC yet. Atomic force microscopy was nice, very nice visually to be able to see this, the ARC as a monomer and then possibly as a dimer. So looking at the surface of the ARC protein. So these two domains are very different. It has an N-terminal domain, it's highly basic, so it's very likely to then interact with polyionic surfaces. The C-terminal domain we think is the oligomerization domain. It's acidic and stabilized by salt, leads to this oligomerization. We believe that is the oligomerization domain. And then, yes, I'm getting ahead of myself a bit here. Let me see. So we know then that it has these two domains, and a number of post-translational modifications have been identified, and some of these are by us and that we're still working on. ARC is known to undergo sumoylation. We see that it's phosphorylated. As I mentioned, it's also ubiquitinated. An important recent finding is the partial crystallization of the ARC C-terminal domain. This is by the Paul Worley lab. And fascinatingly enough, he was able to confirm bioinformatic predictions that the ARC protein evolved from a retroviral capsid protein, the GAG protein. So if you look at the 3D structure, the crystal structure, what you can see is that there's a really beautiful overlap between the structure of the HIV GAG protein and the C-terminal domain of ARC. And uniquely in vertebrates, this piece right here, so-called end lobe, has a binding pocket, a hydrophobic binding pocket and a peptide binding groove. And he's shown that stargazin, which regulates apireceptor trafficking, for instance, docks right in there. So if you mutate residues here, you can block that interaction. So it's an important, functionally an important domain for the ARC protein. The full crystal structure, we don't know yet. We're trying to get that. And it's also interesting that some of these partner proteins that I mentioned that were mutated in the context of schizophrenia risk, that they bind within this pocket. So it's a single pocket, potentially then interesting in terms of small molecule modulation. There have been a lot of successes recently in the use of small modulators for just this type of effect. And so where we are now really is that we're getting an idea of what sort of switches may exist on ARC, and then we're pursuing those. I don't have a full story to report on. Hopefully next time I will have that. But these are really the possibilities: conformational changes due to the floppiness of the ARC protein, the post-translational modification, the monomeric versus the oligomeric form of the protein. And so what we'd like to try to do now is to develop a FRET biosensor that potentially could read out the different functional states of ARC related to LTP and LTD, and to put ARC under optical control by light-sensitive proteins, light-sensitive dimerizers. And to do this, then we put together a team, and this was just recently started up. It's funded by the Norwegian Research Council. And these are my partners, the structural biologist, Petri Kursula, the molecular geneticist and protein person as well, Jan Horvik, and then another structural biologist and biophysicist, Aurora Martinez. So we're bringing a variety of techniques to bear, and yes, I think I will close there. I'd like to say just a brief something, make a short comment about precision neuroscience. And you know, that is that you know, I have to agree that we don't. I feel that that most of the treatments, what we have to offer is getting better and better, obviously all the time. But I have to agree with Tu that we really don't know, and Karel, that we don't really know enough yet. It just makes it very difficult. We don't really know how plasticity, these different types of plasticity, are actually being used in circuits in the brain. And the tools really aren't there at the synaptic level yet. For doing circuit analysis and turning neurons on and off, but not really showing what happens to the synapse during a learning experience, during sleep, how is the plasticity process during sleep, and what have you. And then tracing the molecular pathophysiology. So if you have a mutated ARC-interacting protein, there's a possibility here to then explore more in detail exactly how the mechanism goes wrong. And there are potentially opportunities here also for the use for the development of small molecule therapeutics. So thanks for your attention. This is our lab. People who have done all the work, the lab members, and I'd like to thank our collaborators as well on the translation work, Claudia Bogny, Chris Proud, Nachum Sonnenberg, the protein team, you've heard about them, and welcome you to Norway next time.